Hello friends, today we will study about the second biomolecule, that is proteins. These proteins are consisting of amino acids, and the amino acids are joined together by peptide bonds, that is CONH linkage. There are 20 different type of amino acids. Besides that, there are some non-protein amino acids like ornithine, citrulline, diaminopimelic acids. These amino acids do not undergo movement in electric field because due to its neutral charge. Deuteric ions, and these amino acids bind themselves by peptide bond and form the structure proteins. The protein structures are primary, secondary, tertiary and quaternary. Most of the amino acids are levorotatory and some are dextrorotatory. This is the proteins, and proteins are the heteropolymers of amino acids. It is not homopolymers, it is the heteropolymers of amino acids. These amino acids are joined by peptide bonds, that is CONH linkage. Most of the amino acids are levorotatory. Amino acids do not undergo movement in electric field due to its neutral charges. Most abundant proteins in animal kingdom, it is collagen, while the most abundant proteins in biosphere, that is rubisco, ribulose, biphosphate, carboxylase, oxygenase. The proteins have 4 different structures: primary, secondary, tertiary and quaternary. Okay, the primary structure, it is the sequence of amino acid in polypeptide chain. Primary structure is the sequence of amino acid in polypeptide chain. This primary structure determines the specificity of the proteins but does not make it functional. That means the primary structure determines the specificity of the proteins but does not make it functional, because for functional 3D structure is essential, and this primary structure do not show any 3D structures, so it does not make the functional of the proteins. Secondary structure, alpha helix plated sheets, especially beta plated sheets. In alpha helix, the chains are arranged like coil. In plated sheets, 2 or more chains are joined together by hydrogen bond. In alpha helix, it coil formations. In beta plated sheets, they are bind together by hydrogen bonds. Tertiary structure. Here, the polypeptide chain bend and fold. In tertiary structure, the polypeptide chain bend and fold that make it globular shape, compact globular shape. And here different type of bonds are there: ionic bond, hydrogen bonds, covalent bonds, van der Waals interactions. The best example of this tertiary structure is the hemoglobin, globular shapes. The fourth one, that is quaternary structures. Here the aggregations of the polypeptide chain by hydrophobic interactions. Here the aggregations of polypeptide chains by hydrophobic interaction. Besides this, it also contains hydrogen bonds, ionic bonds. Like the best examples are the blood proteins, maximum blood proteins, especially acidic proteins. In this way, the primary structure, secondary structure, tertiary structure and quaternary structure. The primary structure is a simple chain having specificity to the proteins but not determine the functions, because for functions 3D structure is essential. Secondary structure, alpha helix, beta plated set. In beta plated is antiparallels. Tertiary structure, polypeptide chain fold and bend. In quaternary structure, their interactions, polypeptide chain interact by hydrophobic interactions. When the denaturation of proteins takes place. What is the meaning of denaturation of protein? Denaturation means it has 4 characteristics. Generally 3 characters. One is extra character. First character, the protein lost its functional activity. The protein lost its functional activity. If the protein lost its functional activity, then we said denaturation of the protein. Second thing, the 3D structure of the protein changed. We can also call it denaturation of the proteins. Third, weak non-covalent bonds broken. H bond, hydrophobic bonds. These are the non-covalent bonds. We also call it denaturation of the proteins. Denaturation proteins does not means the breakdown of peptide bond. That means in denaturation of proteins, the 3D structure change, the non-covalent bonds become broke, weak non-covalent bonds break, and the functional activity change, but the peptide bond cannot be broken. This is the important one. The peptide bond never broken. Two different type of proteins: simple proteins, conjugated proteins. In simple proteins, it is this protein, small globular molecules. Small globular molecules. Soluble in water. Soluble in water. The size of the protein molecule is inversely proportional to its solubility. If the size of the protein molecule increases, the solubility comparatively decreases. The fibrous protein, it is the longer protein molecules, so it is insoluble in water. Best example. The fibrous protein is the longest molecule and it is insoluble in water. Conjugated proteins. Conjugated protein means simple proteins with prosthetic group. If the prosthetic group is nucleic acid, then it is nucleoproteins. If the prosthetic group is metal, metalloproteins. If pigment, chromoproteins. The best example of nucleoproteins, protamines. That is protamines. Metalloproteins, hemoglobins. Chromoproteins, cytochrome. Here we are discussing two questions related to proteins. The first question, that is, variation in proteins are due to. Variation in proteins are due to. First option, sequence of amino acids. Second, number of amino acids. Third, R group. Fourth, none. The change of variations in proteins is due to, answer will be the sequence of amino acid. This is the answer. That means the sequence of amino acid is responsible for the variations in proteins. The second question, which of the following is least involved in stabilizing 3D folding of most proteins. Protein folding, 3D folding. Options are hydrophobic interactions. Option 2, ester bonds. Option 3, hydrogen bonds. Option 4, electrostatic interactions, van der Waals interactions. Question asks that which of the following is least involved? If the question will be which of the following is involved, that means hydrophobic interactions responsible for folding, yes, in tertiary structures. Ester bond, it is the covalent bonds. Hydrogen bond also involved. Electrostatic interaction also involved. But ester bond is least involved, less involved, because covalent bond is less involved in folding, especially, and these are the more involvement. So the answer will be because it is a covalent bond, and its involvement for 3D folding is comparatively less or least in comparison to others. In this way, the proteins chapter is completed. Thank you.